surprising findings that we had was discovery of plague in the middle of the 14th century. Chronicles and other historical sources did not predict plague at that early stage.
What we found out was that the genetic diversity was the highest in the early Middle Ages. It's a completely other way of looking to history.
Ancient DNA offers a unique window into the past, complementing traditional history. In Sint-Truiden, researchers Toomas Kivisild and Maarten Larmuseau used advanced genetic techniques and VSC's supercomputing power to uncover 1,000 years of ancestry, migration, and community life, bridging genetics, archaeology, and history to reshape our view of Belgium's past.
We have chosen Sint-Truiden because it was really a coincidence that we were looking for a big cemetery that gives us a lot of information about the past, and that the city council decides to restructure the whole green market or the groenmarkt in city center. So that was really a very nice idea to combine a new archaeological excavation and our new techniques with ancient DNA.
Sint-Truiden material is unique in a way that it goes through more than 1,000 years as a transect of time, and it is a large cemetery, and using ancient DNA allows us to use it as a time machine to go back in time and to check what happened in different time periods. And using the teeth of the individuals, we can track not only the human population historic events, but also the pathogens, the bacteria and the viruses that infected humans at different time points.
What we found out was that the genetic diversity was the highest in the early Middle Ages. It's a completely other way of looking to history.
One of the interesting discoveries was the finding of plague in the middle of the 14th century, which was surprising because the local chronicles and other historical sources did not predict plague at that early stage. We extracted DNA from the remains of more than 400 individuals, and we sequenced the whole DNA, then mapped it to different reference genomes, including the human for human population analysis, and to different bacterial and viral genomes to study the pathogens. We were particularly happy about the support we got from VSC through the course of imputation analysis in particular, where we actually developed the imputation pipelines for the dual usage on modern prenatal screening, low coverage sequence data from the modern cohort as well as on ancient individuals. And the two-tier system of VSC was particularly helpful, as in the tier 1 we could do a number of tests on the imputation efficiency, comparing the tools, different parameter settings, and then later we've been able to use these efficiency optimized pipelines on tier 1 system to generate large numbers of imputed genome. The ability to use the VSC servers was essential for carrying out all the complex bioinformatical analysis required in this project, and by doing so to convert ancient DNA data into accessible information to the historians and general public.
Without the Vlaams Supercomputer Centrum, we would work for ages to combine all these small DNA fragments and to reconstruct the genomes of the past. So thanks to the power of that computer, we can speed up our work.